Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology boys, and this is for 8th October 2020. Our memory helps us in tracing out what we have been discussing. We are into the revision mode, and in the revision we are revising the chapter Principles of Inheritance and Variation. In our last session we were talking about chromosomal theory of inheritance, and we are thankful to two scientists, Walter Sutton and Theodore Boveri. And also we discussed the importance of linkage and crossing over during meiosis. Also we understood how the sex of the baby will be determined not only in human beings, also in birds. So keeping that in our background, we go into the another important concept called pedigree analysis. So what is this pedigree analysis? It is in the form of a chart basically. So it is a chart showing the record of inheritance of certain traits. Traits means characters for two or more ancestral generations of human being or domesticated animals in the form of a diagram with a family tree. What is the use of this? Pedigree analysis is always useful in many ways, like it helps to fill up the possible genotypes by knowing the phenotypes only. It helps to study the pattern of inheritance of a dominant or recessive trait. The possible genetic makeup of a person for a trait can also be known with the help of pedigree chart or pedigree analysis. So it is basically analysis of traits in several generations of a family is called pedigree analysis. The inheritance of a particular trait is represented in a family tree over several generations. It is used to trace the inheritance of a particular trait, abnormality and disease. So we can understand by seeing this, like if you remember our old custom also, whenever you go for a proposed marriage, both bride and bridegroom side, they inquire about seven generations on this side and the other side. The reason is there is a lot of science in that. We may not understand our customs, but our customs have got a lot of science. The seventh generation concept is exactly to know the pedigree analysis. So your mummy and daddy will be doing pedigree analysis without having studied biology. That is the greatness of your parents, right? So pedigree analysis is always helpful in knowing the abnormality. Is there any abnormality in the bride or bridegroom to be? Is there any genetic disease in their family? We'll come to know. So being biology students, you must always keep in your mind that pedigree analysis must be taken. And what are the various genetic disorders that unfortunately some of us suffer with or suffer from? The genetic disorders, broadly genetic disorders may be grouped into two categories. One is Mendelian disorders, that is due to genes, and second is the chromosomal disorders. So the gene related disorders are Mendelian disorders. These are due to alteration in single gene. The Mendelian disorder is alteration. And what about chromosomal? These are caused due to absence. One chromosome is absent, or one chromosome extra present, or abnormal arrangement of chromosomes, one or more chromosomes. That results in chromosomal disorders. They are transmitted into generations through Mendelian principles of inheritance. So like we have got the three principles, law of dominance, law of segregation, law of independent assortment. Through this, the defective gene will be processed from generation to generation. And in the case of chromosomal abnormality, these are transmitted as the affected individual is sterile. Next, under Mendelian, they may be recessive or dominant in nature. Like we said, recessive gene and dominant gene. These are always dominant, chromosomal one. Example, color blindness, phenylketonuria. Okay, this is phenylketonuria, PHENYL. Okay, just a spelling mistake, just take care. And phenylketonuria this is. And in this case it is the Down's syndrome and Turner syndrome. What are these? Let us see. The first one is hemophilia. We go with the Mendelian disorders. Hemophilia. This hemophilia is sex linked, particularly X linked recessive disease in which an affected individual having a minor cut leads to non-stop bleeding. By mistake, if there is any cut, okay, there will be bleeding, continuous bleeding. Person dies due to uncontrollable bleeding. So that is the reason why this hemophilia is also known as bleeder disease. It occurs due to deficiency of plasma thromboplastin, and this thromboplastin or anti-hemophilia globulin, we say hemophilia A, during which the exposed blood does not clot. Hemophilia appears only in human males, which can be transferred to their grandson through his carrier daughter. Now, how is that this is getting transferred? A heterozygous female carrier, so female is a carrier, means one who carries that defective gene, can transmit the disease to their son. The possibility of a female becoming hemophilic is extremely rare because mother of such a female has to be at least a carrier and the father should be hemophilic, unviable in the later stage of life. So how is that the hemophilia is spreading from generation to generation? Now, first and foremost thing is the mother should be a carrier. What do you mean by carrier? See, you can see one X chromosome is normal, another X chromosome has got H. H means hemophilic gene. So the father is normal with XY and the mother having this XH during this meiosis gamete formation. One gamete will have normal X chromosome, another will have defective gene, that is X chromosome having defective gene. And since the father is normal, having X and Y chromosome. So when gametes fuse together to form zygotes, now we are taking the Punnett square method. So here we are taking the mother's X and this one XH, and we are taking this father's gametes here, sperms, X carrying, sperm carrying X, sperm carrying Y. When the fusion takes place, X and X. So this is normal female girl, and this X combines with this gamete having Y, XY, normal boy. So no problem, a normal girl, normal boy. But problem comes when this defective gene, chromosome, X chromosome having this hemophilic gene combines with this, and still girls are lucky. It is only carrier girl. But who is the person having the curse? It is XH combines with Y, it becomes a XY, but this X is carrying unfortunately hemophilic gene. So the boy becomes hemophilic. So this is the unfortunately gift from the mother's side. But of course, no mother wants to give such a gift to the child, but it is all genetics, it's all the gamete formation. So ultimately, what is the percentage? So out of the 4, one normal girl, one normal boy, one carrier girl, one hemophilic boy. So if you see appearance physically, it is again 3:1. 3 are normal, one boy only hemophilic. Whereas you may ask me whether the girl can't become, yeah, there is very rare chance. For that, what is required? The carrier mother should be carrier, and the father should be a hemophilic person, X with H, Y. So this is father's and this is mother's. Now during this process, when you put the checkerboard, Punnett square method, so one is the X, another is the XH from female side, okay, from the mother side. She is a carrier. From the father side, it is XH because he is hemophilic, and another is Y chromosome. Now the combination of this X and XH, what is it? Female carrier, she is only a carrier, okay, and XY, lucky boy, no problem, normal boy, healthy boy, normal. And what about this XH, XH, here comes the first time, very rare occurrence of female becoming hemophilic, girl becomes hemophilic, and of course XH and Y, the boy becomes hemophilic. So again, this girl getting XH is only happens when the mother is a carrier and the father is a hemophilic. Now the question is, who will marry a hemophilic person? That's the question. So it's very, very rare. So boys, we being boys, must be careful. That's why pedigree analysis must be done. And come to second one, sickle cell anemia. It is autosomal. See, hemophilia is sex-linked, X-linked, whereas here it is autosomal linked. It is a recessive trait in which mutant hemoglobin molecules, hemoglobin, where is it? Inside the RBC, right? Red blood cells, undergo polymerization under low oxygen tension, causing change in the shape of the RBC from biconvex disc to elongated sickle-like structure, sickle, kurupi shape. So the defect is caused by the substitution of glutamic acid by valine at the 6th position of the beta globin chain of hemoglobin molecule. The substitution of amino acid in the globin protein results due to single base substitution at the 6th codon. Remember, 6th codon of the beta globin gene from, look at this, what is the change? CAG to CUG, only one nitrogen base is the difference, and person is becoming sickle cell anemic. It is due to inheritance of defective allele coding for beta globin. It results in the transformation of hemoglobin A into hemoglobin S, in which glutamic acid is replaced by valine at 6th position of each of two beta chains of hemoglobin. The substitution of amino acid in the globin protein results due to single base substitution at 6th codon of the beta globin gene from GAG to GUG. So sickle cell anemia is a blood disease where the red blood cell become sickle shaped as compared to normal one. So these are the things, and we are unfortunately, look at this, HBS. S stands for sickle shape, okay, sickle shape, right? And moving ahead, phenylketonuria. It's an inborn error of metabolism, rate at which the digestion takes place, inherited as autosomal recessive trait. The affected individual lacks an enzyme that converts amino acids phenylalanine to tyrosine. As a result, what happens? The phenylalanine gets accumulated and converted into phenylpyruvic acid and other derivatives that results in mental retardation. This is the phenylketonuria. So it is characterized by severe mental retardation, hypopigmentation of the skin and hair, eczema, itching skin, mousy odor of the skin, hair and urine due to increased phenylalanine in blood as well as urine. That's phenylketonuria. And then we come to chromosomal disorders. Of course, in gene-related disorders, there is one more called thalassemia. It is also an autosomal linked recessive blood disease transmitted from parents to offsprings when both the partners are unaffected carrier for the gene. So it is heterozygous. The defect could be due to either mutation or deletion, which is ultimately results in reduced rate of synthesis of one of the globin chains, alpha beta chains, that make up hemoglobin. This causes the formation of abnormal hemoglobin molecules, resulting into anemia, which is characteristic of the disease. So anemic it is. Now we move ahead with the chromosomal disorders. failure of segregation of chromatids during cell division results in loss or gain of chromosomes, and we call this as aneuploidy. And the failure of cytokinesis leads to two sets of chromosomes called polyploidy. Aneuploidy, polyploidy. In this first one, Down's syndrome. How to remember this Down's syndrome? You must remember this as trisomy of 21. Write down trisomy of 21st chromosome. That means 21st numbered chromosome, instead of 2, 3 will be there. That's why trisomy. So it is due to presence of additional copy. See, additional copy of the chromosome number 21. The affected individual is short-statured with a small round head, furrowed tongue, and partially opened mouth. Mental development is retarded. This is Down's syndrome. And then second one, Klinefelter's syndrome. In Klinefelter's syndrome also, there is extra. What is that? It is due to presence of additional copy of X chromosome. There 21st chromosome. Here X chromosome. Instead of XY, there will be XXY. Such persons have overall masculine development. However, the feminine development, like development of breasts, will take place. We say gynecomastia is also expressed, and they are sterile problem. So they are tall, mild mental deficiency, long limbs, and are always at risk of educational problems. So Klinefelter says 46 plus 1x, 46 plus x. And then we come to Turner's syndrome. Turner's syndrome, you must always remember it as 45. So it is called due to lack of one chromosome. There we said extra X. Here it is X0, Turner's syndrome. So in Turner's syndrome, the female with retarded sexual development, usually sterile, short stature, webbing of skin in the neck region, cardiovascular abnormalities, and impairing of hearing impairment. All these are the symptoms of Turner's syndrome. So such females are sterile. So these are the various diseases that we talk of, and one more is there, that's called Edwards syndrome. Edwards syndrome. This Edwards syndrome is trisomy of 18th. Remember 21st trisomy we said Downs. Edwards is 18th. Trisomy of 18th chromosome. So what happens here? Multiple congenital malformations of many organs, receding mandible, small eyes, mouth and nose, severe mental deficiency, congenital heart defects, and horseshoe or double kidney. So these are the various symptoms of Edwards syndrome. And of course, these are the various ways that the Turner syndrome will happen, then Klinefelter's will happen, all that unfortunately during gamete formation. So with this, we are completing the revision of this chapter, and tomorrow I will be talking about the pedigree analysis related to your practicals. Until then, keep practicing.